Okay, so let's continue our discussion of the immune system. And this chapter will be talking about adaptive immunity. And this is going to be primarily the B cells, the antibodies that they produce, and your T cells. Okay, so let's start out by defining some terms. First term I want you to be aware of is immunocompetence. This is going to be the ability of the body to react with foreign substances. And those foreign substances are called antigens. These are going to be molecules that can be seen and identified by the immune system. And if they provoke an immune response, they are going to be called immunogens. Now, there's only a small fragment of the antigen that is actually going to interact with the immune cell's receptor. And that small fragment that binds to that receptor is called the epitope. Next, you have the major histocompatibility complex. I want you to be aware of this. This is a set of genes that is going to code for human cell receptors, okay? And those human cell receptors convey things like self. This is also called the human leukocyte antigen system. So we've got two classes I want you to be familiar with. Class 1. This is present on all nucleated human body cells, and this is going to be the receptor that tells your immune cells, hey, I'm part of the body. I'm normal. I belong here. The second class is going to be found only on antigen-presenting cells. So it's going to be your macrophages, your dendritic cells, and your B cells. And what class 2 does is it's going to help present antigens to the immune system, and we'll talk about that in detail. Remember, an immunogen is going to be any substance that provokes an immune response. An allergen is going to be a substance or an antigen that provokes specifically an allergic reaction. If the foreign molecule is too small by itself to provoke an immune response, we call it a hapten. And these haptens can be bound to larger carrier molecules, like larger proteins. And when they're bound to the larger proteins, the immune system can recognize them. So that combination is going to develop immunogenicity. Next, you have alloantigens. These are going to be cell surface markers that are going to occur in some people, but not in others. An example of this would be blood type. So some people have the A antigen on their blood cells. That would make them type A. Some people have the B antigen on their blood cells. That would make them type B. Some people have both. That makes them type AB. And some people don't have either. That makes them type O. Another example of an alloantigen would be what we just talked about with the major histocompatibility profile. Remember that class 1 is going to be present on all nucleated cells. So the vast majority of body cells are going to have class 1 MHC molecules on them. And these differ slightly from person to person. One of the reasons that we do extensive blood typing for organ grafting and for transfusion is to try to get these alloantigens as similar as possible between two people so that there's less of an immune reaction to them. So hopefully the immune system doesn't recognize them as foreign. Superantigens. Okay, superantigens are going to be bacterial toxins that really stimulate T cells. So you'll learn that normally T cells need another cell to present them an antigen before they can become activated. Superantigens, on the other hand, can directly activate T cells. And in doing this, they're going to activate them at a rate 100 times greater than usual antigens. And then autoantigens. These are going to be molecules on the self cells, for which tolerance is inadequate. An autoantigen is just going to be essentially a regular molecule on the body's cells that the immune system mistakes for foreign and develops an autoimmune reaction to. Okay, so we're going to kind of start with the big picture, the overview of adaptive immunity, and then we'll get into the details of how exactly the immune system works. So we've said before that the immune system is going to be composed of cells, secreted molecules, proteins, and a bunch of different kind of barriers. And together these are going to function to provide protection from pathogens, toxins, as well as abnormal body cells. We've said that the immune system is going to be divided into two categories. You have the innate immune system and adaptive immune system. The innate immune system is going to be cells, barriers, and mechanisms that protect against a broad variety of potential pathogens. These are present at birth. That's why they're called innate. And the response time for innate immunity is going to be fast because these mechanisms are already in place, ready to go. Adaptive immunity, on the other hand, this is going to be cells and defenses that are activated only once the immune system has been exposed to a particular pathogen, a specific pathogen, and this is called specificity. The response time for the adaptive immune system, at least initially, is going to take longer because they have to develop their response over time. But when they're challenged with the pathogen again a second time, the response will be faster. And this is going to result in long-term immune memory. So what cells are involved in adaptive immunity? Well, it's going to be your B cells and your T cells, okay? So over here we have B cells, and these guys are going to be the guys that produce antibodies. Over here we have the T cells. You've got your cytotoxic T cell and your helper T cell. The cytotoxic T cell can go on to directly act with an infected cell or an abnormal body cell and cause it to kill itself through apoptosis. The helper T cell, you'll notice, kind of acts as a bridge between the B cells and the T cells, and it helps activate these guys. It helps get the B cells primed and ready to go, and it helps get the T cells primed and ready to go. All right, where do they originate? Well, B cells and T cells are both going to originate in the red bone marrow. B cells complete their development in the bone marrow. T cells are going to move out of the bone marrow and complete their development in the thymus. That's why they're called T cells, T for thymus. Now, a mature B cell has B cell receptors on its surface, you can see here, that are going to be capable of recognizing antigens. All these B cell receptors are, and we'll talk about this in a little bit, are antibodies bound to the cell membrane. T cells, on the other hand, they're going to have T cell receptors, and then they're going to have a co-receptor, either a CD8 co-receptor or a CD4 co-receptor. If it's a helper T cell, it's going to have a CD4 co-receptor. If it's a cytotoxic T cell, it's going to have a CD8 co-receptor along with its T cell receptor. All right, when we talk about adaptive immunity, we're really talking about two different kinds of responses. We have cell-mediated responses, and we have antibody-mediated responses. Cell-mediated responses are going to be led by your T cells, the cytotoxic T cells and the helper T cells. Now, remember, T cells can only start doing their job if another cell picks up an antigen and presents it to them. And that other cell might be a dendritic cell, a macrophage, or a B cell. Once activated, helper T cells are going to release chemicals that help coordinate and activate the cytotoxic T cells as well as B cells. Cytotoxic T cells, once they're activated, once they receive that signal from the helper T cells and from the antigen-presenting cells, they're going to directly target the infected or abnormal body cell and induce that cell to undergo apoptosis. B cells, on the other hand, are going to exert their effects through antibodies, okay? So B cells can be activated upon direct recognition of an antigen by their B cell receptors. In other words, they don't need another cell to grab that antigen and present it to them. They can find that antigen, they can see that antigen themselves. Once they are activated, B cells are going to differentiate into antibody-producing cells that are called plasma cells. And these plasma cells secrete millions of antibodies into the bloodstream. Now, what do those antibodies do when they're in the bloodstream? Well, they're going to bind antigens. And when they do that, they can do a number of different things with those antigens. They can neutralize them. So, for example, antibodies might cover up a microbe so that it is no longer harmful. They can be involved in agglutination. This is going to be cross-linking or clumping of foreign cells, which makes them easier to be removed by immune cells. They can be involved in precipitation. This is another kind of cross-linking, except instead of cross-linking cells, it's going to be cross-linking particles, such as viruses or toxins. And again, this is going to form kind of complexes or clumps that can be better eliminated by the immune cells. Antibodies can also activate complement. Remember, complement is that group of proteins in our second line of defense that help destroy threats. Antibodies can also activate natural killer cells and macrophages. We haven't really talked much about natural killer cells. Just know that they're an immune cell. They're involved in killing abnormal body cells, okay? And then another thing that antibodies can do is they can act as an opsonin. And all an opsonin is basically a molecular tag. So if you see a pathogen that you want your immune system to eat up, to phagocytose, your B cells might tag them with antibodies, tag that pathogen with antibodies, and that's going to make it easier for the phagocytes to grab hold of that pathogen and engulf it. All right, let's talk a little bit about development and maturation of B and T cells. I expect you guys to know where B and T cells originate. It's going to be in the bone marrow. And I expect you to know where T cells mature. And that's going to be in the thymus. And I'm going to go over what that maturation process looks like. Okay, so B cells and T cells, we said, are going to form and develop in the primary lymphoid organs. Remember, that's going to be your bone marrow and your thymus. Both B and T cells are going to arise from those pluripotent stem cells in the red bone marrow. You got to know that. B and T cells originate in the bone marrow. B cells are so named because they're going to complete their development in the bone marrow. T cells are going to leave the bone marrow and finish developing in the thymus. Remember, T for thymus. Now, once they've finished developing, but they haven't encountered an antigen yet, they're going to be called naive. All right, so those T cells are going to leave the bone marrow. They're going to head to the thymus. Once they get to the thymus, they're going to be called thymocytes. Thymocytes are going to begin here kind of on the outside of the thymus in the cortex, and they're going to migrate towards the medulla, or the inner part of the thymus. And there's a hormone, thymosin, that's going to stimulate this process. Okay, each thymocyte expresses an antigen-specific T cell receptor. What does that mean? Each T cell is going to have T cell receptors that are built to respond to just one particular type of antigen. And there can be infinite number of antigens that our immune system can interact with. And the way that our immune system makes these receptors so that they can potentially interact with an infinite number of foreign molecules is through gene shuffling. And this process is called somatic recombination. B cells also undergo gene shuffling and somatic recombination. Now, the thing to remember is that each T cell is going to have identical T cell receptors on its surface that can only interact with one particular antigen. Okay, but you've got a bunch of T cells in your body, and each one has different T cell receptors on it. So altogether, your immune system can interact with countless antigens. The T cells also have these co-receptor proteins, the CD4 and the CD8 proteins. And these guys are going to start being expressed in the thymus. So when a thymocyte is developing, it actually expresses both the CD4 and the CD8 proteins at the same time. This is called a double positive thymocyte. All right, so here we've zoomed in on a double positive thymocyte. So it's going to have its T cell receptors, but it's also going to have co-receptors. And the double positive is going to have both the CD8 co-receptor and the CD4 co-receptor. These double positive thymocytes are going to go on to these screening processes called positive and negative selection. These screening processes are extremely stringent. Only about 2% of cells are going to make it through. Now, positive selection is going to test the ability of these thymocytes to bind those MHC proteins. And what the question positive selection is asking is, can the thymocyte recognize self? Double positive thymocytes that either have a CD8 protein that binds MHC class 1 or a CD4 protein that binds MHC class 2 are going to go on to survive this process and undergo negative selection. Okay, so here we have a thymic epithelial cell. It's using its MHC class 1 molecule to display a self-antigen. The thymocyte, the double positive thymocyte, is going to bind with its T cell receptor to that self-antigen, and its CD8 co-receptor is going to interact with the MHC class 1 molecule. Now, I want you to make note of this. CD8 co-receptors are always going to only interact with MHC class 1, whereas the CD4 co-receptors are always going to interact with MHC class 2. Now, if the thymocyte can bind to this MHC complex with the antigen, it's proved that it can recognize self. So it's going to pass positive selection and move on to negative selection. Okay, the thymocytes that survive positive selection are going to go on and lose one of those co-receptors, either the CD4 co-receptor or the CD8 receptor, and become single positive thymocytes. And those single positive thymocytes are going to undergo negative selection. Negative selection is going to test the ability of these thymocytes to tolerate self-antigens. That is, it asks of the thymocytes that can recognize self, which ones recognize it too well? Which ones are likely to react to self? So this screening, negative selection, is important in order to avoid producing self-reactive T cells, which can lead to autoimmune disease. Okay, again, in negative selection, you're going to see these thymic dendritic cells displaying self-antigens on their MHC complexes. And the thymocytes that bind with those self-antigens are going to undergo apoptosis because they've proven that they are too reactive to self. All right, so here's a dendritic cell of the thymus. It's presenting self-antigen on its MHC class 2 molecule. So if it's a class 2 molecule, we know that the co-receptor it's going to be binding is CD4. Remember, CD4 is going to bind to class 2. CD8 is going to bind to class 1. This is a single positive thymocyte, so it's only got CD4 receptors and T cell receptors. And you'll notice this is binding very strongly to the self-antigen. So will this thymocyte survive negative selection? The answer is no. It's going to be induced to kill itself, to undergo apoptosis. Okay, so once the thymocytes pass negative selection, they're going to be ready to enter the circulation as naive T cells. Your CD8 positive cells are going to be called naive cytotoxic T cells, while your CD4 positive cells are naive helper T cells. Okay, so CD8 is always the cytotoxic T cell. CD4 is always going to be the helper T cell. The fully developed T cells, as well as the B cells, are going to migrate to secondary lymphoid organs, so like places like the lymph nodes and the spleen. And the reason they do that is that they're likely to encounter foreign antigens in these places. Okay, so this is what those naive T cells look like. On the left here, we have a CD8 positive T cell. So this is going to be our cytotoxic T cell. And you can see it's only going to be expressing the CD8 co-receptors along with the T cell receptors. And note that the T cell receptors on this lymphocyte are all going to be identical to one another, and they're only going to respond to one particular antigen. We go over here, we see our naive helper T cell, and you'll notice it's only expressing CD4 co-receptors along with its T cell receptors. And again, these T cell receptors are all going to be identical on this cell, and they're only going to respond to one particular antigen. The CD8 T cells, remember, are going to bind to MHC class 1, while the CD4 T cells are going to bind to MHC class 2. Okay, so here's a question. B cells mature in the blank and T cells mature in the blank. Both cell types then migrate to the blank and blank. So, recall that B cells are so named because they mature in the bone marrow, and T cells are so named because they mature in the thymus. And then remember, the bone marrow and the thymus are going to be your primary lymphoid organs. That's where B and T cells are going to originate and mature, and/or mature. And then once they're done maturing, they're going to migrate to the secondary lymphoid organs, places like the lymph nodes and the spleen. And the reason they do this is so that they can come into contact with potential pathogens. Okay, so before we get into the nitty-gritty of cell-mediated immunity and humoral immunity, let's just kind of recap what we've been talking about. So, the major role of a B cell or the T cell is to recognize antigens somehow. B cells are going to recognize them directly. They have receptors that can bind antigens. Remember, these are called the B cell receptors, and they're basically antibodies that are membrane bound. T cells cannot bind antigens directly. They can only bind antigens that have been processed and complexed with those MHC molecules, and the cells that do this are called antigen-presenting cells. So T cells can only recognize antigens that have been processed and presented to them by antigen-presenting cells, things like the macrophage, the dendritic cell, and B cells. Once they're activated, T cells are going to go on to release things that are going to kill the infected cell or the abnormal cell through apoptosis. Okay, so they're going to induce that cell to kill itself. B cells are going to exert their actions through the release of millions of antibodies. And remember, those antibodies are going to go on to interact with the antigen in various different ways that we talked about, things like neutralization, agglutination, opsonization, okay? So first we'll talk about cell-mediated immunity, and what I want you to remember about cell-mediated immunity, big picture, is that it's going to involve T cells, okay? And these T cells are interacting with antigens that are being presented to them by antigen-presenting cells. So first we'll talk about helper T cells, how they work, how they get activated. So helper T cell activation, remember, is going to depend on them binding to an antigen that's been presented on an MHC class 2 molecule. It's the antigen presenting cell that does that. Now, helper T cells in particular are going to be able to recognize exogenous antigens. These are going to be antigens that originate from outside of the body cell. So, for example, an antigen presenting cell might engulf an extracellular bacterium, break that bacterium down, and present that exogenous antigen to the helper T cell. All right, so let's look at and see how this works exactly. So over here we have our T cells. We have the helper T cell and the cytotoxic T cell. And here's our antigen presenting cell. Now, our antigen presenting cell is going to engulf this extracellular pathogen, this bacterium. And then it's also going to make MHC class II molecules. And it's going to bring those together and complex antigens from that bacterium onto the MHC class II molecule. Then it sends that complex to the cell membrane where it can communicate with T cells. Now, which particular T cell is going to interact with this processed antigen? Remember, it's only the CD4s that can interact with antigens that have been put on MHC class II molecules. So it's going to be our helper T cell that comes in here to recognize this antigen. All right, and when it does that, you can see both the T cell receptor binding and the CD4 co-receptor binding. So here's our T cell receptor. Here is the MHC class II molecule, and here is that antigen from that bacterium. The T cell receptor is going to interact with the antigen, while the CD4 co-receptor interacts with the MHC class II molecule. This interaction here, this binding, is what's going to activate this helper T cell so that it's no longer naive. So that activated helper T cell is going to go on to release cytokines, in particular interleukin-2 or IL-2. And what IL-2 does is it signals that helper T cell to make a bunch of copies of itself. Okay, and when it makes a bunch of copies of itself, those copies are going to differentiate into either memory helper T cells or effector helper T cells. Now, effector helper T cells are just, what you think of when you think of a helper T cell. The T cell that's going around doing its job, releasing cytokines and interacting with cytotoxic T cells and B cells as well. Memory T cells, on the other hand, are just going to kind of hang around. They don't really do the work that the helper T cells do. They're just kind of on standby for future exposure to the same antigen. And when they get exposed to that same antigen, again, they're going to get activated and they're going to release IL-2, and this whole process is going to get started. So here we have the activated helper T cell. Remember, it's activated because it bound to that antigen presented to it on MHC class II. And when it gets activated, it releases IL-2. And that IL-2 travels to the IL-2 receptors on the helper T cell and stimulates the helper T cell to make a bunch of copies of itself. And these copies go on to be effector helper T cells and memory helper T cells. All right, now let's talk about the cytotoxic T cells, the CD8 positive T cells. So their activation is going to rely on endogenous antigens presented to them with MHC class I molecules. Endogenous antigens are going to be antigens originating from within the cell. So things like a protein fragment from a virus. Remember, a virus is an intracellular pathogen. Okay, here we have our T cells, and over here's our antigen presenting cell. And you can see here we've got some viral peptides inside the cell. So the cell's going to take the peptides from the virus and again complex it with an MHC molecule, this time an MHC class I, because it's going to be interacting with the CD8 T cells. So this complex molecule, the MHC class I and the antigen, gets sent to the cell surface where it can interact with the CD8 cell. that CD8 cell is then going to bind to that presented antigen. And you'll notice it does it much the same way that the CD4 or the helper T cell does. So you've got the T cell receptor that's going to interact directly with that antigen that's been presented and complexed on the MHC class 1 molecule. But you also have that CD8 co-receptor coming down, and it binds to the MHC class 1 molecule. Again, it's this binding that's going to activate the T cell. And so the cytotoxic T cell then releases interleukin-2, and that again is going to cause it to make a lot of copies of itself. You'll note that interleukin-2 is also going to be released by nearby helper T cells. So these copies are going to go on to differentiate into both either memory cytotoxic T cells or effector cytotoxic T cells. Okay, and those effector cytotoxic T cells, they're the ones that are going to go on to destroy the unhealthy cell, while the memory cytotoxic T cells are going to hang out and be ready for a future encounter. So we said that effector cytotoxic T cells, just like the helper T cells, contain those T cell receptors on their cell membranes that are specific for just the antigen that caused their activation. Cytotoxic T cells, once they're activated, they're going to go on a hunt. They are hunting any body cell that displays the antigen that caused their activation. So they're looking for infected cells or otherwise unhealthy cells. Okay, but how does the cytotoxic T cell actually cause the unhealthy cell to die? Well, that cytotoxic T cell is going to bind to the infected cell, bind to that antigen on the infected cell, and then it's going to release these things called perforin and granzymes, which are going to signal that cell to self-destruct through apoptosis. Okay, so here we have an infected cell, and it's displaying the antigen on its MHC class 1 molecule, right? So this is an endogenous antigen. And the cytotoxic T cell recognizes this antigen and binds to it. And upon this binding, it's going to release these things called perforins, which create a pore in the unhealthy cell, and granzymes, which slip through that pore and get inside of the unhealthy cell and signal that unhealthy cell to undergo apoptosis. Okay, so here's a question. Which of the following correctly describes the physical interaction between an antigen-presenting cell and a naive helper T cell during activation? A. The T cell receptor binds to the exogenous antigen presented. That's true. Remember that the helper T cells are going to recognize exogenous antigens, while CD4 binds to the MHC class 1 molecule displaying the antigen. This is incorrect. It's going to bind to the MHC class 2 molecule. The answer here is going to be B. Now you should know why the other two are incorrect as well. C, the TCR binds to the exogenous antigen presented. Again, this is accurate because we're talking about a helper T cell, while CD8 binds to the class 2 molecule. Okay, this is wrong for a couple reasons. CD8 is not the helper T cell. CD8 is the cytotoxic T cell. Also, you should know that this is wrong because CD8 never binds to the MHC class 2. It only binds to the MHC class 1. And again, D is talking about CD8 cells, which are not the helper T cells. The question is asking about helper T cells. Okay, another question. Which of the following is not an antigen-presenting cell? Well, you need to remember that the antigen-presenting cells are your macrophages, your dendritic cells, and your B cells, not helper T cells. These guys are not antigen-presenting cells. They need an antigen-presenting cell in order to do their job. Okay, final question here. Cytotoxic T cells, all right, so we're talking about the CD8 positive cells, are programmed to kill all of the following except. So remember, cytotoxic T cells are going to go after body cells. They're going to go after body cells that are infected or that are otherwise unhealthy, such as cancerous cells, okay? So we're looking for A, cancer cells is correct, B, virus-infected cells is correct, and D, foreign cells grafted into the body is correct. Because remember, even though these are not self body cells, they're going to be displaying their antigens on MHC class 1 molecules, and the cytotoxic T cells are going to see that and go, okay, this cell is not self, let's kill it. So the correct answer here is C. Extracellular bacteria are not going to be recognized by cytotoxic T cells. Okay, let's move on to humoral immunity. Now, humoral immunity is going to involve the B cells, and it's going to depend on the action of antibodies. Okay, so what exactly is an antibody? Well, an antibody is a Y-shaped protein that's able to bind antigen. It has two main regions. The first is this region here, the stem region, or the Fc region. This is the region that's going to determine the antibody class. So, for example, whether it's an IgA antibody, an IgG antibody, etc. Then there's these two arms up here in the variable region, and this is where the actual antigen binding occurs. Okay, just a little bit more detail on antibodies. The two arms of the Y, we said, are going to be the region that binds antigen, and they're called antigen binding fragments, or Fabs. And at the end of each Fab, there's basically a very complex groove that accommodates the epitope, or the fragment of the antigen that actually interacts with the antigen binding site. The rest of the antibody is going to be called the Fc region, or the crystallizable fragment. It has a pair of heavy chains that are identical to one another, and a pair of light chains that are identical to one another. And these guys are bonded through disulfide bonds, and I'll show you that right now. So here we have our variable region where the V is. This is going to be unique to each an antibody. So, for example, a B cell will have a whole bunch of, or produce a whole bunch of antibodies with the same variable region. But another B cell might produce completely different antibodies with a completely different variable region. The C here stands for constant, so this area is the constant region, and this is going to be the region that's the same across each class of antibody. You can see the heavy chains here, and you can see that they are bound together by disulfide bonds. And then you see the light chains out here that are bound to the heavy chain via a disulfide bond as well. And now, this Fc region has binding sites down here at the end that can bind to receptors on cells such as macrophages, neutrophils, eosinophils, mast cells, basophils, and lymphocytes. Why do antibodies have these binding sites for these cells? Well, one reason is for opsonization. Remember, that's going to be when you tag something for phagocytosis. So let's say this antibody interacts with the antigen on a pathogen. Then it goes on to bind to a macrophage via its Fc region. That's going to trigger the macrophage to engulf the antibody-antigen complex, okay? So it's going to trigger it to basically to eat the pathogen. That's one way antibodies work. Another way, if this were, say, an IgE antibody, this Fc region binds to basophils and mast cells and causes them to release their granules, which include allergic mediators such as histamine. So IgE is involved, the immunoglobulin involved in the allergic response. Okay, recall that we mentioned that some of the functions of antibodies are going to be neutralization, agglutination, or precipitation. Now, during neutralization, the antibody is going to bind an antigen that's actually on the microbe. So here you have a bacterium, and it has antigens that it's displaying on its cell surface, and you have these antibodies that are binding to those antigens and effectively covering them up so these antigens cannot interact with our body tissues. An example here might be lipopolysaccharide or endotoxin that essentially gets cloaked by the antibodies so that our body does not interact with it. Another way that antibodies can work is through agglutination. So this is going to be the cross-linking of foreign cells. Okay, so this is going to cause them to clump together, and that's going to make it easier for our body to remove them. Antibodies can also cross-link smaller particles, such as viruses or toxins, and this is, again, going to cause them to fall out of solution or precipitate and be more easily removed by immune cells, and that is called precipitation. Okay, and once that antibody binds the antigen, remember that there's that stem region of the antibody that is able to go on and interact with other immune structures. It can go on and activate complement through the classical pathway. It can activate natural killer cells. Natural killer cells are going to work much like cytotoxic T cells in that they're going to induce apoptosis through perforin and granzymes. And don't forget that stem region can bind to a phagocyte, and in that way induce phagocytosis, and this is called opsonization. Okay, here's opsonization. So you have your antibodies that have bound antigen that's attached to this microbe, and that stem region, the Fc region of the antibody, goes on to bind to receptors on the surface of the macrophage, and that's going to initiate the phagocytosis in this macrophage. You can see it forming its pseudopods, and it's about to engulf this microbe. Okay, we've talked about helper T cell activation. We've talked about cytotoxic T cell activation. Now let's talk about B cell activation. And B cell activation is going to be a little bit different, remember, because B cells have those B cell receptors that can directly bind to an antigen. They don't need antigen-presenting cells in order to respond to antigens. In fact, B cells are themselves antigen-presenting cells. What happens is their B cell receptor is going to bind to that antigen, and that's going to initiate the B cell to undergo endocytosis, or bring that antigen in, process it, and present it with its MHC class II molecules. So that means it's then going to present that antigen to a helper T cell. And the helper T cell goes on to produce cytokines that activate the B cell. Okay, so over here we have our naive B cell, right? It has not been activated yet. And over here we have an activated helper T cell. Now the B cell recognizes an antigen. It binds to its B cell receptor, and then the cell undergoes endocytosis and brings in that B cell receptor and antigen. And then it takes that antigen and processes it and complexes it with an MHC class II molecule. It sends this processed antigen and MHC class II molecule to the cell surface where it can interact with the helper T cell. And remember, the helper T cell is going to bind to that antigen with its T cell receptor, and its CD4 co-receptor is going to bind to the MHC class II molecule. Now when that happens, this helper T cell is going to release cytokines such as IL-4. IL-4 is going to travel to the IL-4 receptors on that B cell, and that's going to send a signal to the B cell to activate. And when it activates, it's going to form a lot of copies of itself. Okay, so an activated B cell proliferates, that is, forms many copies of itself, and these copies are called clones, okay? And those clones are then going to differentiate into either memory B cells or plasma cells. I want you to know, plasma cells are the B cells that secrete antibodies. Memory B cells, again, they're just going to hang out. They're on the ready for when the body encounters this antigen again. And these memory B cells are ready to proliferate and produce plasma cells on re-encounter with antigen. Now, the secreted antibodies are going to all be identical to one another, and they're all going to target that very same antigen that activated the original B cell. Okay, here you have an activated B cell, and it's going to proliferate and differentiate. So it can differentiate into either a memory B cell, and this guy is just going to circulate, hang around, and wait for interaction with that antigen again in the future. Or it can proliferate and differentiate into plasma cells. And these plasma cells are the B cells that are the antibody secretors. All right, here's a chart going over the different antibodies, and I do want you to be pretty familiar with this chart. So I'm just going to walk you through it. First, let's talk about IgG. This is a monomer, so it's only it's found as a single antibody, a single Y-shaped protein. I want you to know that IgG crosses the placenta. Its Fc region is going to bind to phagocytes, so this is going to be the guy that's involved in opsonization. Okay, and then down here, I want you to know this stuff in bold. I want you to know that IgG is the most prevalent antibody that circulates throughout the tissue fluids and the blood. It's going to be the guy that neutralizes toxins, opsonizes, and it's going to activate complement as well. And don't forget, it can cross the placenta. Next, we have IgA. This is found as both a monomer, right, a single Y-shaped protein, or it can also be found as a dimer. And that's going to be two antibodies kind of bound together. Now, I want you to know that the dimer is secreted on mucous membranes. So IgA is going to be our big defender in the respiratory tract and in the gut, okay? It's the secretory antibody. Now, IgM is a huge molecule. It is a pentamer. So it's going to be 5 antibodies bound together. What I want you to know about IgM is it's going to be the first antibody that's made in response to an antigen. And we'll talk about this in a second. But when you look at immune responses, when you look at antibodies produced, there's going to be basically two stages of the response. A primary stage, which takes a while because it's the first time these B cells have seen this antigen, so they're having to make antibodies from scratch. And then there's a secondary response. This is the second time your body encounters an antigen after all those B cells have already been primed and seen it. And the secondary response is much faster because those B cells are ready to go and unleash their antibodies the moment they encounter that antigen. Well, IgM is going to be produced in the primary response, the first stage, because it's the first guy out of the gates. It's the first antibody produced in response to a given antigen. Another thing it can do is it can also trigger complement. Okay, next is the IgD antibody. This guy's a monomer, and it's the B cell receptor. Okay, so just think of IgD as being that receptor that's on B cells. And then finally, IgE. I want you to know a little bit about IgE. I want you to know that it's the antibody involved in allergic reactions and also in parasitic worm infection. So what IgE is going to do is it's going to bind that antigen, and then it's going to have its FC portion bind to basophils and mast cells. And those guys are going to release their granules, which are predominantly inflammatory mediators like histamine. And so that's really the underlying mechanism to allergy, is this FC portion of the IgE antibody binding to those mast cells and those basophils, causing histamine release. Okay, here's that primary and secondary response I was just talking about. So let's look at the chart down here. We have here on the y-axis is the antibody titer, and a titer is just the level of a given antibody in the serum. And so we have this red line, which is going to be our IgM antibodies, and we have this light blue line that's going to be the IgG antibodies. Okay, and this dark blue line is the overall or the total amount of antibodies in the serum. So let's go over the primary response first. Here we have first exposure to the antigen. So this is the first time the B cells have ever seen this antigen. Let's say it's from a microbe. And so they are getting activated. They are differentiating into memory cells and into a bunch of plasma cells. And those plasma cells are making their antibodies. This takes time, and the time it takes is called the latent period. So in this interval of time here, no antibodies have been released yet because they're still being made. Now, when antibodies are released, the first antibody out the gates, we said, is going to be IgM, and you can see this spike here in the IgM levels. Then the IgM is going to wane, and the IgG is going to come in. So it takes a little bit longer for your B cells to produce IgG than it does to produce IgM. Okay, and then we get over the infection and everything's okay. Then a variable amount of time goes by. This could be days, weeks, months, years, okay? And the body sees the antigen again. So this is second exposure. This is going to be our secondary response. And you can see the body makes IgM again, but it's not the first guy out. It's released at the same time as IgG. And look at all this IgG that's being released. And this is because those memory B cells, the memory B cells hang around, and when they get exposed to that antigen, they already have antibodies made that they're ready to just pour out into the bloodstream. Okay, so that's how the secondary response happens. Okay, here's a question. Match each antibody type with its description. So let's start here with IgG. And what did I say you needed to know about IgG? Well, I want you to know it's the most prevalent antibody that circulates. So Let's see if that's one of our answer selections. Here is D. So IgG is the most prevalent antibody. Number 2, IgA. Remember, we said IgA is going to be secreted on mucous membranes. So A for IgA. IgM, remember, we said this is going to be E, the first antibody produced. IgD, all you need to remember about that is that it is the B cell receptor. And then IgE, the important thing I want you to remember about IgE is that it is produced in response to allergies, and this is going to be the guy involved in allergic reactions. Okay, there's a couple of other immune cells I just want you to be aware of. You don't need to know anything specifically about them, but I just want to mention them. I want you to know that there are regulatory T cells. These guys kind of apply the brakes to the immune response. They control the T cell response by secreting anti-inflammatory mediators or by preventing proliferation. So they keep that T cell response in check. I've also mentioned before the natural killer cells. These guys are actually a part of the innate immune system. And what they do is they patrol the body, they identify, and they destroy infected, or especially, you might know them as the cancer-killing cells. And what they do is they recognize abnormal surface markers, and they release cytotoxic substances like perforins and granzymes. They can also activate B and T cells, so they act kind of as a bridge between the innate and the adaptive immune system. And then a little word on helper T cells. There are various types of helper T cells. There's the Th1 helper T cell, the Th2 helper T cell, and the Th17 helper T cell, among others. But these are kind of the three primary ones you may hear mentioned. And they're going to secrete slightly different cytokines to activate different cells. For example, the Th1 helper T cells activate cytotoxic T cells. The Th2 cells activate B cells. And Th17 helps promote inflammation. Big picture here that I want you to remember is that helper T cells, remember, they're going to become a bridge between the humoral response and the cell-mediated response. They're the guys that activate both B cells and T cells. So they're vitally important in the immune response. And you need to know that helper T cells are the targets of HIV and AIDS. They are disproportionately affected. So helper T cell counts are going to drop dramatically in patients with AIDS. And this is the reason why we see immunodeficiency in them. The T cells are crucial for both the function of B cells and of cytotoxic T cells. Okay, now we're going to get into vaccination. But before we do, we need to define a few terms. We need to define natural versus artificial immunity, and we need to define active versus passive immunity. Natural immunity is going to be any immunity that's acquired through the normal biological experiences of a person, okay? Artificial immunity is going to be protection from an infection obtained through medical procedures. Active immunity is going to occur when a person receives an immune stimulus, okay, that activates their B and T cells, and immunologic memory is formed. Passive immunity, on the other hand, is going to occur when an individual receives antibodies from another human or animal. This can be natural or artificial. We'll talk about it in a second. But no immunologic memory is formed. Now, you can have natural active immunity, or you can have natural passive immunity. And then you can have artificial active immunity and artificial passive immunity. Natural active immunity is going to be any immunity acquired through the normal biological experiences plus the individual receives an immune stimulus. So this is going to be like a normal disease. The patient just gets sick, the body builds antibodies to that, and the immune system is activated the old-fashioned natural way. That's natural active immunity. Natural passive immunity is going to be an immunity, again, acquired through normal biological experiences, but no immunologic memory is going to be formed. So they're going to receive antibodies passively. And the example for this is breastfeeding. Mothers pass on antibodies through breast milk. So baby gets protection from mom through breastfeeding. Now, artificial immunity can be artificial and active. So this is going to be, an example of this would be vaccination. Artificial means that we did some sort of medical procedure, and active means that the person is able to form an immunologic memory. That example is vaccination. An example of artificial passive immunity, again, remember, artificial means it's some sort of medical procedure, and passive means there's not going to be any memory formed. An example of this would be antibody infusion. Okay, so let's go into a little bit of depth over artificial active immunity, or vaccination. So the basic principles behind vaccination: you want to stimulate a primary response and a memory response. And you want this memory response because you want to prime the immune system for future exposure to that pathogen, so that when the pathogen enters the body's response is immediate, powerful, and sustained. Vaccines have profoundly reduced the prevalence of infectious diseases that were once common and deadly. Okay, let's talk about the qualities of an ideal vaccine. So an ideal vaccine is going to protect against exposure to the natural wild forms of the pathogen, right? That makes sense because that's the form of the pathogen you're likely to encounter. They're going to have a low level of side effects, low toxicity, and not cause harm to the person receiving the vaccine. Ideally, a vaccine would stimulate both B cell and T cell responses. That's not always the case, but ideally that's what you want. Ideally, you want long-term lasting effects. That is, you want the vaccine to produce immunologic memory. You want the vaccine to not require a bunch of doses or boosters. And you want it to be inexpensive, last for a long time, and be easily administrable. Okay, so vaccines can be prepared in a few different ways. They can use the whole cell or the whole virus when preparing the vaccine. And the cell or virus can be either living or active, or dead or inactive. If it's alive, it is attenuated somehow. That means that they have weakened the pathogen so that it's not pathogenic. It's not going to cause disease. Another way vaccines can be prepared is by using antigenic components or subunits that are derived from the cell or virus. And these are called subunit vaccines. So you can get subunits a variety of different ways. You can get them derived from cultures of cells or viruses, or you can get them as synthesized to mimic natural molecules found on pathogens. But the idea here is that this is going to be a piece of the antigen or a piece of the pathogen that's going to stimulate an immune response. Now, sometimes the subunits by themselves are not very immunogenic. That is, they don't really elicit a very strong immune response. So they'll take the subunit and they'll complex it with bigger proteins, and that is going to make it more visible to the immune system. These are called conjugated vaccines. So if you've ever heard the term conjugated vaccine, it's a type of subunit vaccine. You can also have vaccines that contain DNA or mRNA. And remember, these guys are going to code for something. And in vaccination, they're going to code for microbial surface components. So antigens. And what we do is we put these molecules inside a lipid vesicle using nanolipids. And these lipids are going to help this molecule cross over into our cell membranes, where these molecules can be made and displayed on our cells. And then our cells see the antigens and create an immune response to it, create antibodies to it. Another way we can do it is we can use a viral vector. That is, we take a harmless virus and we have it carry a DNA or an RNA, again, for some sort of antigen from the pathogen, and we infect ourselves with that virus. And so our cells start making that DNA or that RNA, that molecule, that antigen is going to get made and displayed on our body cells, and our immune system is going to respond to it. So that's kind of the logic behind vaccination. Okay, here we have the whole virus or the whole cell vaccines. So on top here is the killed cell or the inactivated virus. So we've got a microbe here, and it has antigens on its cell surface. We apply heat or chemicals or something to kill that microbe, but the antigens remain intact. And then we inject this into our body, and our immune cells react to these antigens. An example of this would be the injectable flu vaccine. On the bottom here, we have an example of a live attenuated cell or virus. So here again, we have the microbe and it's displaying its antigens. And then we have somehow reduced its virulence or eliminated its virulence. So it's still living and it still has its antigens. But when it gets injected, it's not going to cause the disease. Now, these guys are still living, so they can multiply in our body. And the multiplication is going to basically stimulate more immune cells, and that's going to lead to a stronger immune response ultimately. So live attenuated vaccines can result in smaller doses and fewer boosters needed. Now, one risk to the live attenuated vaccine is that theoretically these pathogens can mutate back to their virulent strain. It's really mostly theoretical. It's extremely rare when this happens, but when it happens, they can cause disease. And so for that reason, we don't give live attenuated vaccines to people with who are immunocompromised, okay? And we don't give it to pregnant people because they can theoretically pass the microbe onto the fetus, which could theoretically get the disease. Okay, now let's look at subunit vaccines. So a simple subunit vaccine here would take something like a surface antigen from a virus or a bacterium and inject that antigen into the body, and the immune system is going to react to that antigen and build a response to that particular antigen. An example here would be the hepatitis B vaccine. Another way to do it would be to take that surface antigen, complex it with a protein to make it more visible to the immune system, and then inject that into the body. And that's an example of a conjugated vaccine, and that's going to induce a stronger immune response. Examples here would be Haemophilus influenzae type B conjugated vaccine. mRNA vaccines. So here we have a piece of mRNA that when it gets put into the host cell is going to be translated into the viral protein or the protein of the bacterial antigen, whatever it's encoding. Okay, this is encoding some kind of antigen. And it's bound inside of this lipid vesicle. And we inject those lipid vesicles into the body, and the lipid vesicles cross the cell membrane and drop that mRNA transcript off. And our cells translate that mRNA and create that antigen and display the antigen on their surface. And then our immune system reacts to that antigen. An example here is the Pfizer and Moderna COVID-19 vaccines. Okay, and then finally, we can use a viral vector. So we take a third-party virus that doesn't cause any disease, and it's got a DNA or an mRNA inside of it. And this DNA or mRNA is going to code for the antigen. So we inject that virus, it infects our cells, our cells start making that mRNA or that DNA, and then it produces that antigen and displays the antigen on the cell surface, and our immune system responds to that antigen. Example here is the Johnson & Johnson COVID-19 vaccine. Okay, how are vaccines administered? Well, most are going to be administered either subcutaneously, intramuscularly, or intradermally. However, there are a few nasal and oral vaccines out there. Now, they're only available for a few diseases, and they're going to stimulate IgA in particular. And this makes sense, right? Because IgA is the immunoglobulin of our mucous membranes, and we're taking these vaccines through our mucous membranes. The great thing about these is that they're so much easier to give than injections, and they're more readily accepted by patients, especially children. All right, some vaccines have adjuvants. These guys are going to enhance the immunogenicity of the vaccine. And so think about it this way. It's going to, they can help the antigen stick around longer. So either at the injection site or be released gradually from the tissues. And that's going to mean that there's going to be more antigen around to interact with immune cells over time. Adjuvants can also facilitate contact with APCs and with lymphocytes. And some can also help involve the innate immune system in the response as well. Now, some vaccines wane over time, and so booster shots need to be given. Example here would be tetanus. You need to get your tetanus booster every 10 years. And measles has a booster at about the age 5 or 6. Now, COVID-19 required booster shots, and the reason for that is because the SARS-CoV-2 virus had a very high mutation rate. So it was basically outpacing our vaccines at the time. So we had to develop boosters to address new strains of COVID-19. Okay, side effects. Vaccines, by and large, are safe. They are safe to administer. They go through years of trials in experimental animals and then human trials before they are licensed. However, there are some complications that can occur. Local reactions at the injection site, fever, allergies, Guillain-Barré syndrome has been linked to some vaccines. However, more serious reactions are going to be extremely rare. By and large, vaccines have little or no side effects, or their side effects are manageable. Now, people have attempted to link vaccination to development of several diseases, including, of course, autism. And that goes back to 1998, when Andrew Wakefield published a scientific paper linking autism to vaccination. However, the paper was later discredited. He falsified the research, And for this, he actually had his medical license revoked. So there is no current evidence to suggest that vaccination and autism are linked in any way. Okay, final question here. Determine which of the statements below describes active or passive immunity. And I'm going to go a step further, and I'm going to ask, is it natural active or natural passive, or is it artificial active or artificial passive immunity? Okay, A, infusion of gamma globulin. Okay, this is an antibody, and we're infusing it, so it's a medical procedure. So this is going to be artificial. And infusing antibodies is not going to elicit an immune response from us. So no immunologic memory is formed. Therefore, this is passive immunity. So artificial passive. B, recovery from influenza. Well, this is a natural process. So it's not a medical procedure. So it's going to be natural. And then it's using our normal biological response. We're creating an immune memory from influenza. So B is going to be natural active immunity. C, receipt of the influenza vaccination. Well, this is a vaccine, so it's a medical procedure, so it's going to be artificial. And it's stimulating an immune response. So we're going to produce immunologic memory. So it's active. So vaccination is an example of artificial active immunity. And I want you to know that for the exam. D, antibodies passed from mother to infant through breastfeeding. Okay, well, it's antibodies. Which are not going to elicit an immune response from us. So no immune memory is formed. So this is going to be passive. And it's breastfeeding. This is a natural biological experience. So this is going to be natural passive immunity.